Okay, so this is maybe something a little different from some of the other presentations. Um, so this is an artwork that's currently in progress that I'm going to talk about. And the focus is on pattern forming bacteria. Um, these bacteria were discovered by or at least their pattern forming behaviors and their swarming behaviors were discovered by Eshel Ben Jacob and others in the 1990s. Um, we've been working with these for a few months now, and what we're doing is we're creating an artwork that is essentially sort of a hybrid, machine-microbial artwork, right? That features a bio-driven AI system that remediates a toxic soil ecology while generating an audiovisual composition in real time. So the idea is that it creates a situation where the fate of the contaminated soil and the fate of the bacterial cultures are determined by sort of like the whims of the AI. And the AI has an internal model of beauty. So that's the basic idea. So it observes the pattern forming behaviors of the bacterial cultures and tries to sort of alter them and shape them so that they sort of match the model of beauty. Now, the sort of hook here is that these bacteria, they grow like these patterns in a sort of environment. So they don't grow like this in the natural environment. These are when you put them on agar plates. That's what they do. And they sort of get these patterns in kind of low nutrient environments. So the idea here is that they have to starve themselves to look beautiful for the AI. And if they do so sufficiently, then it'll remediate that toxic soil ecology. And the way we're doing it is sort of altering the bacteria is we'll have a system that sort of drops into the plate certain stress-inducing chemicals, essentially, like basically attractants and repellents. So stress-inducing chemicals like antibiotics, not enough to kill them, but to stop them from growing or changing them in some way. And other things as well. We're looking at transforming them so they can generate different colors as well. So it's sort of, you can think of the AI as kind of like a I don't know, a film director or a painter that's sort of altering these bacterial growth patterns. So quick background on the bacteria themselves. So these plates here that you see are Bacillus subtilis, one of the species that we're working with. So this is how they grow in different concentrations of peptone. Peptone is their food essentially. So this is they're grown on agar plates at a certain concentration of agar, and then these different concentrations of the food will give you different patterns, right? I mean, if it's too concentrated, you get like that first one on the top left where you don't really get any patterns. So the system is basically this, right? You have a machine learning model, which I'll talk about in a second, sort of maintaining or altering the bacterial cultures. We use time-lapse microscopy or video, and then that goes back to the machine learning model, right? So the idea is that they'll sort of reduce nutrient levels or induce, like I said, stress-inducing chemicals, et cetera. And as this is going, so it's like a real-time thing. As this is, let me show you the sort of larger image, poster-like image. And as this is going, we're also looking at sort of maybe determining, for lack of a better term, the health status, and then sort of visualizing it and sonifying it. So the idea is that the AI will express its feelings about what's going on by generating sound and vision. So so this is kind of the overall system diagram. And this is how it might look sort of in an installation setting. So the model is, we're using, for right now, we're basing it on what's called the world model, which is a deep reinforcement learning model. So basically you have three components. You have an autoencoder, a recurrent neural network, and then the controller, the reinforcement learning controller. So the autoencoder, does what an autoencoder does, which is give you. So that's the eyes, right? So it outputs so what it sees as a temporally and spatially compressed representation of the environment, the image of the plate. So that's your latent vector. And then the M here, the MDN-RNN, mixture density network, recurrent neural network, that's your predictive model for future Z vectors, right? So that the autoencoder is expected to produce. So it's trained to output some probability distribution about the next latent vector. The way they describe it in the paper, is that it's basically dreaming of other possible environments, right? Over time. And then, the controller then is responsible for actually taking action, right? So it maps the Z vectors and the hidden states directly to actions. Now, what's unique about this model is that you can actually train it completely within its own generative model, right, of the environment instead of the environment itself. So, and then that's why they call it dreams. It's sort of hallucinating of possible environments, and then you sort of essentially transfer that to the controller. So that's where we're at right now, sort of collecting data and working on the stock world model. But ideally, we'd want to have some more sort of domain-specific knowledge, sort of like the physics of the growth and the pattern forming of these bacteria. So deep learning, as I was saying here, it's sort of kind of like a black box, right? And typically, I mean, the idea is to not have to do much work with your data, right? You just sort of throw it data, and typically you sort of ignore scientific principles, right? So in this context, adding a physics domain model, which is kind of a new thing that they're doing. Willard et al. They've been using it to model the physics of water dissipation in lakes and that sort of thing. The idea is that'll give you a more robust model where it's hard to get good, clean data, right? So you have sort of a little bit of this domain-specific knowledge to sort of help it out. And fortunately, Ben Jacob and his collaborators, they made some sort of basic, models of these bacteria. They have essentially two basic models. There's a few variations on each, but there's basically two. The communicating walkers model is a discrete, kind of like a grid-like model of where the bacteria essentially do a sort of random walk. The cells are they're called walkers, and each cell really represents, I forget, several thousand bacteria, but they're in a continuous field of these sort of chemicals in the agar, and an explicit sort of free boundary of the colony edge. And then the lubricating bacteria model is a continuous model. It's essentially it's very similar to diffusion limited aggregation, which, as you saw from the images, that's kind of how those patterns look. Some of them, not all the bacteria look like that, but so that's that one. It couples bacterial movement to a kind of this field of this lubricating fluid. And both models include, like food diffusion, sporulation, all these kinds of things. And then the aesthetic evaluation, right? Like, what is that model of beauty? We're just gonna. This is the hard part in my view. So we're just going to start with something simple like fractal dimension. But, it's probably a lot of this is a real sort of big challenging computational creativity. So, but it is sort of known that visual aesthetics and complexity, they're related, right? And I would add other, not just visual, but there are known to be related. So, so that's, we have a lot of work to do there, but we're starting with this kind of fractal dimension. So I'll basically leave it there, for questions, and I'll just sum it up. Just say this is like a kind of a speculative, very conceptually driven artwork. So it's sort of based on this kind of interplay between, we're sort of thinking about, sort of the potential for these kind of AI hybrid microbial systems as, kind of media generation systems. But even this project itself, it's, mixing a lot of things, cultural notions of beauty and aesthetic judgment, the status of intelligent machines, ecological issues, all kind of mashing them together in this kind of, so as as an artist, I feel these things are related. So that's kind of the motivation. And I'll just finish up by saying this is supported partially by the Coalesce Center for Biological Art and the University of Buffalo in Buffalo, New York, in the U.S. So we're working there on the bacteria stuff. And this is part of a group project where we're known as Phylum. We're an experimental research collective working at the intersection of science, technology, and the arts. So feel free to visit our website or email me with any questions. Thanks.
Thank you very much, Carlos. I am very grateful to you because I saw that it's very late at night there or very early in the morning.
Yes.
So thank you. Thanks a lot for doing this effort, for being here. And also thank you very much to everybody attending very hard times around the world. So thanks to all of you. Regarding your work, well, this is definitely a bio-inspired approach, of course, and I like the generative art papers. You know that there is a conference inside EvoStar just devoted to evolutionary music and arts.
Yeah, of course.
So it's Evo Muse Art. So
yeah,
I hope you will send a work next year to this conference. And regarding your work, it's very interesting, very attractive, let's say. Do you? Well, I don't know if is there any other question. It seems there is not. So, what's your plan with this research? I mean, once you tune your algorithm, your algorithm is working properly according to some evaluations. This needs to involve some human people, I guess. What do you plan to do with the results?
Yeah, so right now, I mean, it's in its very early stages, as you can tell, and the data collection is slow just because the nature of this kind of work. You have to do it at the rate at which the bacteria grow. So, our first goal would be just to build the stock model and kind of just see what happens. And then just sort of just build on that. But this is really kind of a big project with many mini projects, like other installations and performances. But yeah, I mean, ultimately it would be to exhibit it as an artwork.
Exactly. It's very nice. But I'm wondering if is there a project devoted to this kind of research, right? You're working in a project for doing this research specifically.
I'm sorry, could you repeat that? Is there a what?
Are you participating in a project for doing this specific research?
Well, this is, I mean, I presented the artwork, but yes, this is part of a kind of a larger kind of research track I'm into, and what I'm calling bio-driven compositional agents is actually, creating these hybrid systems to
nice.
like these can we use these patterns as and the swarming and other things that I haven't even talked about, can they be harnessed with AI models to develop kind of hybrid systems that can, a big thing in kind of this field is expanding the space of creative possibilities with evolutionary algorithms, for example, right? So can we sort of do something with these that you can't just do in software, right? So it's part of that kind of larger research program. Yeah.